Looking in the stomach at the tissue that makes it up, the important part to look at are the digestive glands and the mucosa. And the inner surface of the stomach is fairly, I don't know, flat, I guess you could say, kind of like this. Probably not quite as much as this cartoon kind of suggests, but when you look at the tissue under the microscope, you'll usually see mostly this kind of appearance in 2 dimensions. But really what you're looking at mostly are the infoldings of the mucosa down into these glands. The opening of the gland is called the gastric pit. So just looking at the surface of the stomach, you see all these pits in there. So those are the gastric pits. And then deeper down inside, we have the gastric glands. What's not pointed out in this picture, but it's worth mentioning, the cells that make up the neck of the gastric pit here are pretty much the same as the cells that make up the lining of the stomach. And a lot of them secrete the basic mucus solution. Now, when you hear mucus, you usually think of a nasal mucus, you know, snot. But this is a much more watery mucus. And what's important in this mucus is that it secretes components which help to protect the lining of the stomach from the gastric juices, which we'll look at in a second. So in this inset here, they're sort of highlighting the different types of cells that are in here. So one type of cell that's left out are the mucus-producing cells, which secrete the watery secretion that everything else is dissolved in. These cells, which look kind of funny in shape, are called parietal cells. They are the ones that make the stomach acid itself. And then the main type of cell here is the chief cell. I had to stop for a second and say the chief cells there are the chief cells. But the most common type of cell in the gland are the chief cells, which make the enzyme secretions, which are mainly pepsin, actually pepsinogen, which as soon as pepsinogen is exposed to the low pH of the stomach, it converts to pepsin, which breaks down proteins, which I mentioned earlier. There needs to be that kind of switch from a pro enzyme to an active enzyme, because if pepsin were present in the chief cell in its active form, it would digest down the chief cell. So the enzyme is only active at the low pH in the lumen of the stomach. So that helps protect the actual tissue of your stomach from being digested by that enzyme. And then the last type of cell down here, which is labeled the enteroendocrine cell, secretes an enzyme, a hormone that affects activity in the stomach. Now, if you notice the chief cells, there are all these little yellow dots representing vesicles in what would be the apical surface of the cells, because they're secreting, they're releasing their contents through the apical surface into the lumen of the stomach. In the enteroendocrine cell, there are little dots along the basal surface representing the vesicles that are secreting the hormone made by these cells out into the surrounding connective tissue. That hormone, which is called gastrin, which just means stomach hormone, when it's secreted from these cells, will bind to receptors on the neighboring cells in the gastric glands, as well as into the muscle layers in the tissue, stimulating them to be more active. So it's sort of a signal from the stomach to itself telling it to be more active. And we'll look at some of that regulation in a second. I want to talk a little bit about the parietal cells here. And actually, I was doing this earlier today with one of my other classes, and I wasn't recording. I was just writing it up on the board. So I didn't think about needing to find a good picture of this. So let me just... cell, see if I can find one real quick. This should, oh, no, actually I want to go to this one. No, maybe I do like this one. Nope, they're both kind of complicated. So this one's a little less complicated. So let me go with this. Oh, shoot. I'm sorry. No, that leaves out an important part of what I want here. Nope. All right, this should do the trick. Yeah, this kind of hits everything. And this is not as overly confusing compared to what I was just pulling up. But it's not as nice a looking picture. Okay, so this thing that's drawn here, which somebody must have done on a computer with a mouse, so it looks really ugly. And I apologize to whoever might have actually done that, but yuck. This is supposed to represent this cell here. Under the microscope, you wouldn't be able to tell that the shape of the cell is what's indicated here. You actually have to look at it at the ultrastructure level, meaning with an electron micrograph, to really see that. Under the light microscope, it's going to just look like any one of these other cells here. Just a simple columnar cell. But it actually does have a little bit of a weird shape to it. Now, what's going on in this cell is carbon dioxide and water are being combined into carbonic acid, which is not actually represented in this picture. Instead, it goes directly to showing hydrogen ions and bicarbonate ions. Okay? So I talked about the carbonic acid reaction last week a little bit, and let me just remind you of that more completely. Carbon dioxide plus water gives us H2CO3, carbonic acid. Now I put a double-sided arrow there because this reaction can run either direction depending on what you have more of. If you have more CO2 than carbonic acid, then you're going to form carbonic acid. If you have less CO2 than carbonic acid, then you're going to break down the carbonic acid. And then the carbonic acid reaches an equilibrium between the state we see there and the hydrogen ion and bicarbonate ion components. So in the picture that I'm pulling off the internet real quick here, they leave out the actual carbonic acid in the middle here, which is fine because that's not the point of what we're going at here. So carbon dioxide and water are the raw materials that give us hydrogen ions and bicarbonate ions. Now the hydrogen ions are pumped out of the cell through an ATP-dependent pump, which is a hydrogen potassium pump, which should sound very familiar to you. What does that sound like that you've learned about in the past? Hydrogen potassium pump. Sounds like a...
Sodium potassium.
Yeah, sodium potassium pump. So this is just a variation on that. It pumps hydrogen ions out of the cell and pumps potassium ions into the cell. When it does that, hydrolyzing ATP. Now, if I were depicting the same thing, I'd add to this that there's a potassium leak channel in the membrane here. So the potassium gets pumped in and then it just leaks right back out. So the potassium isn't really necessary to what's going on here for what's going on in the cell. It's just the pump has to have potassium so that hydrogen gets pumped out. Okay. So the potassium just gets turned around and reused through this. And so as that hydrogen ions pumped out, the pH of the solution that's being pumped into, the lumen of the stomach, is going to go down as the concentration of hydrogen ions goes up. Okay. Now, bicarbonate is produced by this HCO3 with a negative charge, and that leaves the cell through a protein. Was that me or somebody else? Anybody hear a beep? All right. Just want to be sure it's not the browser here making noises. Anyways, so the bicarbonate leaves the cell. And when bicarbonate leaves the cell, chloride enters the cell. Now, this is not an ATP-dependent pump. This is just a anti-transporter protein. And the only way that bicarbonate can leave is if chloride comes in. Oh, there's the potassium leak right there. I would have put it over here. The chloride that gets pulled in when the bicarbonate goes out then just leaks out of the cell. And so we have in the lumen of the cell of the stomach hydrogen ions and chloride ions, which make up hydrochloric acid. Now, the cell never actually takes hydrogen and chloride and puts them together for HCl. But when you have HCl and you put that into water, the hydrogen ions and the chloride ions dissociate and you get a solution of hydrochloric acid. So really the cell is building a solution of hydrochloric acid. The chloride is kind of secondary to that. It's really just there because the bicarbonate leaves the cell. Chloride has to come in. Now the bicarbonate, when it leaves the cell, heads for the blood, which is going to need the bicarbonate to help buffer the pH of the blood. This is very similar to what we saw in the red blood cell last time. In the red blood cell, the same kind of reactions are taking place, helping to transport the carbon dioxide in the blood. And bicarbonate is made in that process. And that bicarbonate from the red blood cells also getting out into the blood, helping to buffer the pH there. Okay, so as far as the bicarbonate is concerned here, from the parietal cell in the stomach and the red blood cell, which we saw in the respiratory system, the same thing, endpoint is coming. Just in the parietal cell, the hydrogen ions end up getting pumped out into the lumen of the stomach, where they'll contribute to the pH of the stomach contents. In the red blood cell, the hydrogen ion attached to hemoglobin when oxygen left it. Okay, so what's going on this side of the reactions is different between the parietal cell and the red blood cell, but what's happening over here is actually pretty much the same. Okay. And that's how stomach acid is made. It's just pumping hydrogen ions and chloride ions, well, pumping hydrogen ions out and allowing chloride ions to move down a concentration gradient, and they get into the lumen of the stomach, and we get hydrochloric acid. There's a rather popular or successful class of drugs which block this ATPase right here. By blocking this, it keeps the hydrogen ions from being pumped out of the cell, which lowers the acidity of the stomach or raises its pH. People that have gastrointestinal reflux or gastric ulcers will take these drugs to block that pump. It's a very interesting drug in that it has a very, very low side effect profile. It basically only blocks that pump. And it doesn't actually do that good of a job at blocking that pump, which works out great for the effectiveness of the drug, because when you don't have food to digest, you block the pump. When you do have food to digest, the drug is not effective at blocking the pump, and you make stomach acid. So it blocks the pump while you're not needing acid in the stomach to digest food. When you do need acid, the production of acid from these parietal cells overcomes the drug's ability to block this. Can anybody tell me the name of those drugs? Probably heard them before. Either if you've got, I was going for brand name, but yeah. So Prilosec or Nexium are brand names. Omeprazole's the generic name. As a class, they're called proton pump inhibitors because they inhibit this proton pump. A hydrogen ion is a proton, so just blocking that. And somebody in a drug company must have just hit pay dirt the day they discovered that, because they're very successful drugs. They have very low side effect profile, and people buy them all the time because it's a great way to deal with chronic stomach acid issues. They are provided over the counter, Prilosec OTC. They don't require a prescription anymore. The problem is that when you block that, you often are hiding the root cause of the problem. So if you are a person that takes Prilosec OTC on a regular basis without consulting a doctor, just understand that you might be masking a bigger underlying problem by treating the symptom without thinking about the cause of it underlying. And that might be stomach ulcers or something like that. And if they get bad enough, eventually they're going to cause really big problems. But the drug is affecting that right there. So that's a depiction of what's going on in the parietal cell, represented here. The chief cells are just secreting an enzyme. The endocrine cells are secreting a hormone in opposite directions, but they're both doing secretory things, which we've studied before. Another name for the enteroendocrine cells can be G cells, G standing for gastrin. So if you see reference to a G cell, that's what it's talking about, that kind of cell right there. The last thing to consider with the stomach, in the stomach portion of the book, is getting at what's controlling stomach secretions, which is what this figure is getting at. Now, there's three phases of what regulates gastric secretions and really, to some degree, gastric motility. The first phase is what's called the cephalic phase, which means what the head is doing to regulate what the stomach's doing. The red arrow up top is getting at the things that lead to stomach activity. So anything that has to do with sensory systems, meaning special senses in the head, so the sight, taste, smell of food, or even just the thinking of food, which is not a sensory thing, but is going on in the head too. Any of those things will stimulate the hypothalamus, the medulla, to send signals out along the vagus nerve, which will stimulate stomach secretory activity, as the book says here, but also it might increase the motility of the stomach muscles. If you're walking that way across campus and you cross the street, what do you walk past? Over there. Burger King, right. That's a great example of cephalic phase activity. Okay, when you walk past a Burger King or a fast food place of any sort, what are you usually going to be assaulted with, sensorily speaking? What? Yeah, smell. They usually pump like the smell of the fries out or something like that. Activating the smell receptors is going to cause signals to come down through the vagus nerve, telling the stomach to be ready for food coming. Now, you're probably just walking by the Burger King to go somewhere else, like you parked your car over there or something like that. And you're not actually planning on eating. But your brain's like, hey, food, let's eat. And it starts activating the stomach. That's often going to result in what we think of as hunger, just the grumbling of the stomach and those sorts of things we're going to associate with eating. And so we think of that as hunger. And what Burger King has done is by pumping the smell out, it makes you think you're hungry, even though you're probably not hungry, and you'll go inside and you'll get some food. So any of those sorts of things will work. The sight or thought of food is pointed out here, which goes really well with television commercials. Okay, so you're sitting there watching television, and a commercial comes on showing you these amazing pictures of, the best food you can imagine. you start thinking about how much you want that. You start getting hungry. You go get some food. Okay. You're not really hungry, but that's just part of the cephalic phase. The point of the cephalic phase is, marketing practices aside, if you're about to eat, you're going to see the food in front of you. You're going to smell the food in front of you. When you put it in your mouth, you're going to taste that food. So all of those things are leading up to eventually getting the food to your stomach. So the cephalic phase is about preparing the stomach for the food that's going to get there. Okay. Now they mention sort of an inhibitory side of this, which is actually kind of, let's call it minor, not to offend anybody that might have some of these issues, but being the cephalic phase, thinking about higher processing within the cerebral cortex, there are some things that can inhibit activity. So anything that leads to loss of appetite, which might be depression or something like that, could just be, you're watching television and you've just seen the 300th commercial for, I can't think of anything right now, I'm kind of hungry, but whatever commercial just really disgusts you. And it just turn a switch from seeing and thinking about food and going, oh, I'm hungry, to seeing and thinking about food and thinking, I don't want any. That's going to inhibit the activity there. Okay. The next phase is called the gastric phase, which means what the stomach does for itself. So when the stomach stretches out, which is what distension means, stretch receptors are activated, or when there's rising chemical levels, whether it's hydrogen ions for pH or macromolecules that are present in food, any of those things will lead to more activation. The stretch receptors will initiate what are called vagal responses, which is the stomach sending a signal up the vagus nerve to the medulla, which will just pull in some more cephalic phase activity. But then also there are local reflexes that'll cause an increase in secretion and, again, muscle activity too. The point there is, if you've got food in your stomach and your stomach is getting full, then the stomach is going to start being active to get rid of that stomach by digesting it down. And part of that is the churning thing I was talking about before. With each wave of churning, a little bit of the food moves out of the stomach and onto the small intestine. Okay. When nutrient molecules in the food or the pH start to rise, chemoreceptors are going to activate the G cells, which are the enteroendocrine cells that I mentioned before, to secrete gastrin into the blood. And that gastrin, it doesn't actually have to get into the blood, just in the local environment, will again increase activity. Now, going back to this picture here, the gastrin is released into the surrounding tissue. It might get into the blood in the mucosa or submucosa, and it can travel around to other parts of the stomach. So yeah, sure, gastrin coming from the G cell right here is going to tell its neighboring cell to make more enzymes, or to tell the parietal cell in the neighborhood to make more acid. But also the gastrin levels, as they increase, will spread throughout the whole stomach and increase stomach activity there. Okay. So that's what this part down here is talking about. Now, as far as the gastric phase having an inhibitory side to it, the real thing that causes inhibition of activity is the pH. So up here, pH activates G cells to secrete gastrin, which increases secretory activity. But if that pH gets to be excessive to something less than 2, so a pH of 1 is a very acidic solution, then the acidity is actually going to switch gastrin secretion down, which will help to limit the activity of especially the parietal cells making more acid. Okay. They add in here to the gastric phase emotional distress too, which isn't, I think, entirely appropriate as far as gastric phase, because this is something that's happening systemically. I'd say that this is more in keeping with the cephalic phase here. But what they're getting at is in the fight or flight response, when the sympathetic system's active, it's going to override the normal parasympathetic controls. A lot of that's happening within the tissue of the digestive system, but it's not really the gastric phase so much as just the interaction between sympathetic and parasympathetic inputs to the stomach. Ultimately, it's coming from somewhere else. So I'd really say more appropriately that it's more like the cephalic phase. But if you think of it as coming from the spinal cord, cephalic doesn't entirely make sense. So I think they're putting it in here with gastric phase because it's sort of happening at the level of, well, the abdomen, if not just the stomach. The last part down here is the intestinal phase, which is how the intestine feeds back to what's going on. Now, as they've set this up, there's an arrow representing the increase in secretion and a bar at the bottom talking about the inhibition. With the intestinal phase, the inhibition is actually a bigger part of what's going on. If the duodenum starts to stretch out, which it doesn't want to do, it's going to send signals back to inhibit gastric activity. This mentions a few things that play into that. And then if there's an increase in the amount of fatty or other partially digestive foods or stomach acid showing up in the duodenum, which is going to be paired with distension as things are moving into the duodenum. The intestines are going to release a number of different hormones, which are listed down here. There's secretin, GIP, which stands for gastric inhibitory peptide, cholecystokinin, which we usually abbreviate CCK, and then vasoactive intestinal peptide, which we abbreviate VIP. So secretin, GIP, CCK, and VIP are hormones that are released by the intestines that can have an effect inhibiting gastric activity. There's really only one aspect that's at all stimulatory, which is as the stomach empties out, there will be a little bit of an gastrin that comes from the intestine that feeds back into the stomach. I'm not terribly familiar with intestinal gastrin, like I know more about the stomach gastrin, but I was always under the impression, and I might very well be wrong about this, that intestinal gastrin actually inhibits activity. But what's here could be the truth, and I'm just not that familiar with gastrin. With the intestinal phase, it's more about what's described down here in the inhibition stuff. So your head basically dealing with the anticipation of food arriving in the stomach, and then the stomach actually dealing with the food when it gets there, are mostly about activating activity. There are a couple of things that can be inhibitory for the both, but mainly the cephalic and gastric phases are mainly causing gastric activity, whether it's secretion or muscular activity. The intestinal phase is mostly about the inhibition, because as the stomach is active and it's pushing food down into the duodenum wants to slow it down because it doesn't want all of that food coming in at once. It wants to slow down the stomach so just a little bit is introduced at a time, and that can be processed through the duodenum before it goes on to the rest of the structures. So this is kind of a picture of the things that play a role in regulating gastric activity from the head, from the stomach itself, and from the intestines, and looking at stimulatory and inhibitory aspects of both. Just remember the first two are mainly stimulatory and the last one's mainly inhibitory, although the picture doesn't entirely make that point clear. So a lot of what's covered here at the end I've kind of already mentioned. The mucosal barrier helps to protect, has aspects of it protecting against the pH of the stomach, and anything that breaks down that barrier chemically or physically will usually lead to problems of acid burning through some of the tissue there. And then the mechanical digestion stuff is mostly about that churning thing that I talked about. And then the chemical digestion is mostly about breaking down proteins. Okay. So that's the stomach there. The next part of the the next part of the book is getting into the intestines. What I want to do is talk a little bit about the structure of the intestine and then go on to the next part and then come back to finish up talking about the intestines. So the intestines are responsible for absorbing the components of the food that we eat. And that's mostly done because of an increase in an increase in surface area of the small intestine. And there's sort of 4 ways small intestine increases surface area. The first thing is that the length of the small intestine makes a big difference. So let me go to this picture here real quick. This is showing the entirety of the small intestine. Here's where it comes off of the stomach, and here's where it attaches to the large intestine. So this is the beginning and this is the end. If we just had a straight tube going from the stomach to the large intestine right there, it would be a pretty short tube. But in fact, the intestines wrap around the inferior surface of the stomach and then fold over back and forth on each other within the abdomen, making up all of this tube. The distance from the end of the stomach to the beginning of the large intestine might be, say, 10 or 15 centimeters, but the actual length of this tube is much longer. Now, sources that describe the actual length of that tube vary a little bit, I think just because you have to measure that, of course, either during gastrointestinal surgery, where the surgeon's more concerned about the patient than getting accurate data about the length, but they can sometimes run through the whole intestines, especially if they're trying to find tears or lesions or something like that, somewhere along the length of it. So you can run through it that way. It's just the data collection isn't primary in the surgeon's mind. The other way to do it is with a cadaver. It's just when you treat tissue in a cadaver, it changes its properties a little bit. When you fix it with formaldehyde, it'll tend to shrink or something like that. When it dehydrates, it changes its properties too. All of that's to say that there's not a very reliable number that I've come across for what the actual length of the small intestine is. Most sources will probably throw out a number like 7 meters long. But I read something a couple of years ago that suggests that it might actually be shorter than that. A lot of the estimates of its length have been incorrect, but still it's fairly long. The other estimates are maybe in the neighborhood of 5 meters in length or something like that. Now in the book here, they talk about the duodenum, the first part here, as being, they say 25 centimeters. I'm used to saying 15, but maybe I get inches and centimeters confused there. So it's that first part right there. So we got 25 centimeters to start with. And then the next part is the jejunum, which is the green part. And here in the book, they say that that's 0.9 meters, okay, or 90 centimeters, right? Yeah. So, okay, we're up to 1.1, 1.2 meters in length. Okay? And then the last part, which is, I guess, kind of a maroon color there, is the ileum, which is, according to the book, about 1.8 meters long. Okay, so according to the book, they're saying it's about 3 meters long. Like I said, a lot of sources you might find will say something more like 7 meters long, but that seems to be a gross exaggeration of the real length. The 3 meters here probably might be fairly accurate. I haven't found a definitive source that tells me one thing or another. Other sources I found say maybe in the range of 5 centimeters, but 5 meters. But the point being, the difference between a straight shot here and the length of this is pretty significant. So the length is going to increase the surface area just because the surface area is the diameter, sorry, not the diameter, the circumference of the tube times its length. So if the circumference is, say, 7 centimeters around, and you multiply that by 15 centimeters long for a straight tube, that's a fairly small surface area. But if you multiply it by, where we were with that? 3 meters in length, then it's a significantly greater surface area. The second way it does that is through what are called circular folds. Now, there's a Latin version of this which you may come across, and I want to introduce you to it, which is plici circularis. I might have misspelled that. It might not be ES on the end. It might be something else. But circular fold is the English equivalent to that and is what we usually use. And in fact, it's what the book uses. So this figure is showing actually the next 3 things. And what I'm talking about here are the circular folds, which is really kind of the inner surface is scrunched up a little bit on top of the wall of the small intestine. I used a book at one time that had a actual picture of cadaver tissue sort of cut open like this. And the inside surface really kind of looked like, scrunched up fabric, if you can kind of picture that. Here it is in a micrograph. So the mucosa, which is sort of bent over folds of the submucosa, kind of, like I said, kind of scrunches up along the length of the tube. If we were to straighten this out, we would probably get it to be, several times longer than it actually is. And so these circular folds will increase the surface area yet again. So if we went from 15 centimeters in length to 300 centimeters in length with the just with the basic length of the tubes, within the surface of the small intestine, those circular folds make that overall length a lot longer. So if we could flatten out all those circular folds, then the actual inner length might be, 3-4 times, something like that, larger, which is just going to increase the surface area even more. The next aspect are villi, which is plural for villus. This is a sort of finger-shaped extension out of the mucosa, which is represented here, okay, in 2 dimensions. If you could imagine the cells that make up the mucosal epithelium going straight across here, we might fit, let's say, 50 cells from this side to this side. But instead, we have hundreds of cells that go all the way around this. Not actually done the counting. I'm just sort of estimating numbers there. So with the circular fold level of information, level of magnification here, we can see the villi sort of on top of the circular folds. These little finger-like extensions out of the muscularis here, those are villi. And here we can see them closer up. So maybe this one's a good example. We can see a simple columnar epithelium wrapped around this finger-like extension here. Okay. That's a villus. Okay. The next layer of, or the next level of increasing surface area happens at the cell membrane of these cells. So these are the individual cells making up the mucosal epithelium here. And in their apical membranes, there's a folding there into what are called microvilli. So sort of finger-like extensions of the cell membrane sticking out there. So instead of just a flat cell membrane, if that cell membrane's longer, we can have more proteins that transport nutrient molecules across the membrane and into the cell. Okay. This little fringe of microvilli here is also known as the brush border, which I mentioned earlier. There are brush border enzymes like the sucrase, lactase, and maltase, which sit right on the edge of the cells there, breaking the nutrient molecules down their final step before they get absorbed into the cell. Okay. So that fourth level or fourth way that it increases surface area are the microvilli. Again, microvillus would be the singular version of that. Now, I often try to kind of estimate the number of changes that we'd see here, but across these four different levels of increasing surface area, we'd actually see probably many orders of magnitude change. I was doing this morning in my class. Unfortunately, somebody sitting in the front row has a knack for doing math in his head. So he's able to tell me what all the calculations were. I didn't have to figure it out. But we went from, before even considering the length, if we go back here, if we just went from this point to this point straight down, we saw, I think, about an 80 square centimeter surface area. When we incorporated all of the length and the circular folds and the villi and the microvilli, it ended up being, in a very conservative estimate, I would imagine, I think it was 500,000 square centimeters. And in some of those situations, I said, well, let's say this doubles the length, okay? So if you look at these circular folds, really, if we could straighten that out flat, it would probably be 3 or 4 times longer. But in the very conservative estimate I was trying to make, I just say, let's say the circular folds increase the length by doubling it, which is probably not entirely accurate, but that was the estimation. And then it's really hard to estimate what the microvilli mean. In 2 dimensions, you can kind of say, oh yeah, they wrap around and it's that kind of change in length, but they're also going around the circumference that way too. The villi are finger-like extensions, so there's going to just be this whole field of like these villi sticking up. And actually, if you're looking at the tissue with the naked eye, it just has this kind of velvety plush to it, kind of like little fabric threads sticking up out of velvet. I think that's how velvet works. But so these are more than just a 2-dimensional increase in the length. They're a 3-dimensional increase of length and circumference together, and it really expands the surface area quite a bit. But anyways, those are some of the things happening in the structure of the small intestine that lead them to do their thing. Now, so there's the histology stuff, and it describes it all down there. It gets into what the glands are making a little bit down here, and I'm not going to get into that in a whole lot of detail. Actually, I'm kind of getting away from where I want to go. What I want to talk about is something about what the duodenum is actually doing. So we already saw the duodenum has something to do with the feedback to the stomach, regulating gastric activity. But the duodenum is also where the chemical digestion is going to start in the small intestine. The small intestine makes some of its own digestive juices, but also it gets digestive juices from the liver and the pancreas. So in the book, the small and large intestine section we're on right now is followed by the accessory organs. And this section is specifically about the liver and the pancreas, and gallbladder for that matter. So this picture is showing you the liver and the gallbladder and the pancreas, which sit basically on either side of the duodenum, and they both connect to the duodenum. The liver is making bile, and the bile duct from the liver connects to the duodenum, and the pancreas is making its own digestive juices, and the pancreatic duct connects there too. And in fact, I don't think this picture really makes a big point of it, but the pancreatic duct and the bile duct meet at the exact same location. There's one common opening to those 2 ducts, because usually you're secreting bile and pancreatic juices at the same time, because they're both going to play a role in helping with digestion. Now the liver is made up, it's a huge organ, but down at the microscopic level, it's made up of these repeating units called lobules, which are really just highly vascularized filters. So blood moves through the liver. that blood's coming from the digestive system directly, but really all of the blood of your system eventually works its way through the liver. And it filters stuff out of the liver. Now, the digestive system blood is carrying some stuff from the digestion, which will help to stimulate the liver to make more bile. And then it secretes that bile out along, where'd it go? Oh, I thought there was a picture of this. Oh, that's right. The picture's down here with the gallbladder. So here's the liver, and there are two bile ducts coming out of the liver, one from the right and one from the left, that come together into the common hepatic duct. Okay, that's what's carrying the bile from the liver. The common hepatic duct meets up with the cystic duct of the gallbladder, and then the common bile duct is what's going on from that. So that's carrying the bile down to the duodenum. The bile is made in the liver, and as it's secreted out, it goes down that the hepatic ducts and into the common bile duct. If the common bile duct opening is, well, ending is not open, then the bile isn't released. So it backs up in the common bile duct, and it backfills into the cystic duct and finally into the the gallbladder, which will store that bile until it's needed. Okay? The gallbladder will eventually, when it's stimulated to do so, contract and push that bile out into the small intestine. Bile contains things to emulsify the lipids. The pancreas, we've already studied to some degree for its endocrine function, but it's more importantly an exocrine gland because it's a digestive gland. So there are the little islets that make insulin and glucagon that we talked about before, but most of the tissue are these acinar cells or the exocrine cells that make digestive enzymes. And those digestive enzymes collect in the pancreatic duct, which then carries it out to where the pancreatic duct and the common bile duct come together to secrete things out into the duodenum. So there's a picture showing that particular connection. Along with the enzymes that these cells make, the pancreas also secretes bicarbonate, which will help to neutralize stomach acid as it enters into the duodenum. Okay. So those are the accessory organs and what they secrete, which goes into the small intestine at the duodenum there. And so the bile and the enzymes, as they enter into the duodenum, help to digest the food coming out of the stomach as it passes through everything here. And the nutrients will be absorbed throughout the whole small intestine, as will some of the components of the bile, which get recycled to make more bile later on, until everything eventually moves into the large intestine. There's a valve between the ileum of the small intestine and the cecum of the large intestine called the Ileocecal Valve, which ensures that things only move from the small intestine into the large intestine. We don't want components of the large intestine to move back into the small intestine. Okay, so here's a cecum here. Once the food moves into the cecum, it then passes along the large intestine, where it gets sort of compacted into its final form. Mostly what's in here are the indigestible materials, so that's going to become feces as they end up making their way to the rectum. And as the rectum stretches out, the stretch receptors there initiate the defecation reflex, which pushes feces out through the anus. Now, you're not always in a socially acceptable place to defecate, so you have voluntary control over the external anal sphincter, that's skeletal muscle, so that you can keep the defecation reflex from embarrassing you. But eventually, if you just keep trying to hold it in, it's not going to let you do that. Freud made a lot of money and popularity over talking about our initial learning of how to control that external anal sphincter. Okay, but that's Freud for you, whatever. Now, I just wanted to mention that defecation reflex a little bit. The book should talk about it a little bit more, because when we talk about the urinary system, the reflexes for urination are actually very similar the reflexes for defecation. And I'll talk about that when we get to that part of the urinary system. Here's a similar picture showing the histology of the large intestine, kind of similar to the one of the stomach. And the large intestine mostly has intestinal glands, and the surface looks a lot like the drawing for the stomach that we saw there. These intestinal glands are largely making a mucus secretion, which helps to lubricate the feces as they move through the large intestine. And a lot of the cells in there are goblet cells, which are depicted in this drawing right here of the micrograph. These are cells that are making that secretion. Okay. I've already talked a little bit about the bacterial flora when we were talking about the immune system, but there are bacteria that live in the large intestine, which help break down some of that final indigestible material. We're not getting anything out of that other than maybe the liberation of some micronutrients like minerals and vitamins that we can get. But all of the energy-rich molecules that are still left over in the indigestible material are food for the bacteria that live in our large intestine. And they live there because we provide them with that food source. And in doing that, they seem to offer beneficial functions to our body, which is usually the focus of any activity at commercial. So down here it talks a little bit about the defecation reflex also. Okay. So that gets us to the end of the digestive tract and highlighting some of the functions I wanted you to be aware of. This isn't the end of this chapter. The next section I already talked about, which was the liver, pancreas, and gallbladder thing. And then the last part here is chemical digestion, a closer look, or chemical digestion and absorption, a closer look. I already touched on the chemical digestion part here about the enzyme. So here's that table that I showed you briefly. I didn't look at it very closely. We used a different table for more detail. And it just says a few things about carbohydrate, protein, and lipid digestion. I said earlier why I don't really think that lingual lipase is an important thing for humans, but the book includes lingual lipase in talking about it. And they mentioned gastric lipase here, which is present in humans, just it's not active at the pH within the stomach. And then they only bold-faced pancreatic lipase because, as they say here, it's really the only consequential lipase in the system. So that's where most of the lipids are broken down. This is sort of an overall picture of absorption. We take in liquid in the food that we eat or the drinks that we drink. And liquids are secreted in the form of saliva and the gastric secretions and the intestinal secretions. So this is sort of showing the accumulation of fluid volume in our digestive tract, going all the way down to about 1,400 mils down here by the time we get to this large intestine. That 1,400 mils isn't only what's in the large intestine, but that's the cumulative final amount all the way through, if you add up all the numbers in this series here. We reabsorb that liquid mostly in the small intestine. About almost 8 liters of water per day is absorbed in the small intestine. A little bit more is absorbed in the large intestine, and then the last little bit is a component of feces, because feces do need a little bit of water. If they're completely dry, it would be very unpleasant to try to get that out of your body. So that sort of shows the input and output of water to the digestive tract. Okay, and that's kind of an overview of that. Most of that absorption takes place in the small intestines, a little bit in large intestines. If somebody has an intestinal bug where the intestines aren't functioning properly, a lot less is absorbed, and oftentimes that water stays in the digestive tract and becomes a larger component of the feces, which is what we call diarrhea, of course. And if somebody's having that because they're not absorbing the fluids very well, they can dehydrate pretty quickly. So in that case, taking in fluids, more fluids as dietary input is important so that we can absorb the right amount, even though a lot of it's going to end up being lost in the end there. Here it's talking about carbohydrate, protein, lipid absorption, and this figure here really doesn't get at anything but the lipid absorption, which is kind of weird. But here it's showing lipids are broken down from larger fat molecules into smaller and smaller molecules, which diffuse directly into the cell. Lipids will diffuse across the cell membrane with little to no interference. What I'm kind of surprised about that there's not a similar picture showing absorption of carbohydrates and amino acids is because getting those hydrophilic compounds across the membrane requires transport molecules. And so that might be mentioned somewhat in here, but there's not a picture of it. But what this is getting at with the lipids is the lipids, as they're absorbed in here, get processed through the Golgi apparatus and packaged up into these little structures, which are called chylomicrons. And the chylomicrons move into the lymphatic system. Remember, one of the functions of the lymphatic system is transporting dietary lipids. We see a capillary here sort of going next to the lymphatic vessel. And so if we had other pictures showing carbohydrate and amino acid absorption, those would go directly into the capillary, be absorbed directly into the bloodstream. But the lipids go into the lymphatic system to be further processed before they're ready to be transported through the blood so that they don't cause blockage of circulation as fat embolus or something like that. Just a mention of nucleic acid absorption, because they are there, they get absorbed. And then minerals and vitamins are micronutrients that are a component of the food we eat, and those get absorbed also. And then I just talked about water absorption with that picture up above. So that just hits on some of the other things that I wanted to point out here. That's the end of the chapter. The next chapter is on metabolism and nutrition. And I just want to go into this chapter briefly to talk about a couple of things. Metabolism overall is all of the things that happen in your cells, all the chemical reactions. They can be categorized as catabolic, meaning things getting broken down, or anabolic, meaning things getting built up. So a catabolic reaction would be sugar being broken down to release energy for ATP production. An anabolic reaction would be amino acids being joined together to make a protein chain. But there are a lot more examples of these kinds of reactions. And I have these posters. I used to have them up in my office. I took them down a few years ago when my wife decided to decorate my office because she didn't think that this was as aesthetically pleasing as the picture she wanted to put up there. But as a scientist, I think this is very aesthetically pleasing. This is all of the chemical reactions that take place in your cell, in a given cell. Now, this is a little bit idealized. This includes reactions that take place in any kind of cell. Not every cell of your body does every little thing here, but this is largely all of the chemical reactions that take place. There's a second poster that goes with this, which is showing more of the biochemical or metabolic pathways. This poster is more about the DNA, RNA kind of reactions. So this is depicting a little bit more of some membrane compartments where reactions are taking place, and it's sort of about making DNA and transcribing that into RNA and making proteins, that sort of thing. So the point here with these two posters is really that this is metabolism. This is everything that happens in your body. All I want to talk about for metabolism is carbohydrate catabolism, how we break down carbohydrates to get energy. So in the chapter, it has a section on carbohydrate metabolism, which I'm going to go into in a second. It has a section on lipid metabolism, a section on protein metabolism, and a section on metabolic states and energy and heat and nutrition and diet, which are all fine. Actually, if I have time at the end, I'm going to show you something about nutrition there. But I just want to talk about carbohydrate metabolism. And by this, we really mean how glucose is broken down to make ATP. This is an overview of cellular respiration. Okay. So I did talk about the cellular respiration reaction last week, right? Did I? C6, H12, O6 plus 6 O2s yields 6 CO2s plus 6 H2Os. I put that up on the board last time, right? That's cellular respiration. Now, that doesn't actually all happen at once in our bodies. It's separated out into a lot of smaller, easier to control reactions. But that reaction is kind of simplified, or not simplified, it's kind of expanded a little bit here. Here's glucose. It's a 6-carbon molecule. It's broken into 2 3-carbon molecules called pyruvate. Pyruvate moves on to be attached to a carrier molecule. One of those carbons is released as carbon dioxide. And so this thing here, which has the same basic shape as the pyruvate thing here, isn't really a 3-carbon atom. It's a 2-carbon atom. 2-carbon molecule, which is attached to this carrier thing, which carries it into a series of reactions that make up a cycle called the Kreb cycle or the citric acid cycle. Different sources use different names. The 2 carbons that are introduced to that are combined with a 4-carbon molecule to make a 6-carbon molecule. And one of those carbons is broken off as carbon dioxide. And then that 5-carbon molecule is again loses a carbon to carbon dioxide to be a 4-carbon molecule. And that 4-carbon molecule comes around again to pick up 2 more carbons from the next pyruvate molecule that goes through this transformation here. In that, eventually all of the carbons from the glucose are given off as carbon dioxide. 2 of them come off for each glucose molecule at this step, and the other 4 come off in the Kreb cycle. Okay. And so we get rid of all of the carbons from glucose. As we're doing that, we harvest energy. A little of that energy is harvested at the beginning by breaking down glucose. A little bit of energy comes out of the Kreb cycle, but most of that energy is harvested in the form of electrons, which then are passed along a chain of reactions called the electron transport chain, all of which power ATP production. Okay. Glycolysis, the first part here, takes place in the cytoplasm. And then the pyruvate molecule is modified and goes into the Kreb cycle within the mitochondrial matrix, the inner part of the mitochondria. And then the electron transport chain and the ATP production takes place across the inner mitochondrial membrane, what separates the mitochondrial matrix from the intermembrane space, I think it's called, between the inner and outer membranes of the mitochondria. And through that, we make all this ATP. Now, the next couple of figures kind of get into a bit more detail about what's going on here. I don't want to get it too bogged down, and we're not looking at this in terms of the enzymes that cause specific reactions, but rather glycolysis takes a few steps. So first, glucose is phosphorylated. So a phosphate from ATP is attached to glucose, and then another phosphate from another ATP is attached, and we now have a phosphorylated version of glucose with two phosphates on it. And we have to use two ATPs to get there. And then we break that into two pieces, which are not yet actually pyruvate. But in the process, we go through a few changes where we harvest some energy, somewhat in the form of electrons and somewhat in the form of phosphates that come off, energy that is harnessed as phosphates attached to ATP. And that ATP is made in the end here. We get 4 ATP molecules in the end. We put 2 in to start, we get 4 out at the end for a net gain of 2 ATPs. Okay? And with that, we have some electrons that have been harvested and the 2 pyruvate molecules. Okay? The pyruvate, when oxygen is present in the cell, will then go on to the mitochondrion. Okay? So this is a little summary of the glycolysis thing up above. When oxygen's present, the pyruvate is then prepared with that carrier molecule to go into the Krebs cycle. There are 2 pyruvates, 2 half of each glucose molecules that go through this. And with that, we get some energy in the form of electrons, and we get some ATP out of that. And eventually we get into the Krebs cycle. Now, the Krebs cycle is a series of, I can't remember offhand, 6 or 8 different reactions that keep repeating over and over again as we add 2 more 2 more carbon atoms with each round from the pyruvate. And in that, we harvest a little bit of energy as ATP, but a whole lot of energy as electrons. And those electrons power the electron transport chain, which makes a whole mess of ATP. Okay. Here's a little more specifics about the Krebs cycle, which I'm going to skip over because I don't want you to get bogged down with it, but that's this Krebs cycle. Okay. Here's the electron transport chain and ATP thing. So at this point, the glucose has completely been broken down. There's no glucose left over. All the carbon's been released as carbon dioxide. And what's left over of that is energy in the form of electrons. And those electrons are passed from one protein to the next in the inner mitochondrial membrane. Some of those proteins will pump hydrogen ions across that membrane, building up a high concentration of hydrogen ions on the far side of that membrane. The only way that those hydrogen ions can get out is through this big thing here, which is the ATP producing enzyme. It's powered by the physical movement of the hydrogen ions. And every time hydrogen moves through, it powers the connection of a phosphate to an ADP to make ATP. Okay, so we just keep powering that with all of those different hydrogen ions. Now, the electrons, as they've been passed through this chain, at the end, what they do is they react with the oxygen that's available for this to happen. And electrons added to the oxygen make it possible for the oxygen to react with hydrogen ions. So we have protons, which is another word for hydrogen ion, and electrons and oxygen. 2 hydrogens, 2 electrons, 1 oxygen gives us H2O. We make water. And so the carbon dioxide's already come off in the Krebs cycle in earlier stages. Water comes off here, and we get eventually, for every 1 glucose molecule and 6 oxygen molecules that go into this, we get 6 carbon dioxides and 6 water molecules. So that's kind of a run through of all the steps taking place there. And here, this comes back to that original summary of cellular respiration, but now it's throwing in some numbers for the amount of ATP that's made throughout the whole process. Okay. That's what I want to talk about with carbohydrate metabolism, just the catabolism part of it. This goes on to talk about the reverse of that, which is gluconeogenesis. I don't want to talk about that. So really in here, this is the only part. Now, this goes into some of the detail I would like you to get out of it, except for, don't worry about this detail. I don't care that you know the Kreb cycle that much, just that it's a cycle that repeats over and over again as we add more and more carbon into it, and we harvest energy out of those carbon-rich molecules. Okay. Now, I want to point out, because you have this book, you have access to this information, I want to point out another place to see some of what's being described there. And that goes back into the support and movement section of the book, because that's where you're initially introduced to these aspects of metabolism. In the muscle tissue, I think it's in the skeletal muscle section here. Talks about the structure of skeletal muscles, how they're connected to the nervous system. Oh, not that section. It must be in the muscle fiber contraction part. Because muscle fiber contraction, remember, requires ATP. So in the book here, after talking about how that happens and how the ATP is used, the book then goes on to sources of ATP. These two frames here, the second and third part of this, are really, really simple overviews of what's happening. Glucose broken down into pyruvate, we get 2 ATPs out of that. The pyruvate, also known as pyruvic acid, can get into the mitochondria. When oxygen's present, we give off carbon dioxide and water, and we make a whole bunch of ATP. Okay. The specific numbers aren't mentioned here, but it's a quick sort of overview of those two processes, if you want to go back and see it described here somewhat. Okay. I just brought that up because it's something that comes from A&P1, but here with the digestive system, we want to talk about it a little bit more detail. Again, within the carbohydrate metabolism section of the metabolism and nutrition section. Also in this, I want to just point to the nutrition part. This is a fairly recent representation of our nutritional needs. You might be familiar with the food pyramid, but it's got to be about 5 years ago now. The First Lady was part of an attempt to update the nutrition and dietary information that we share. And part of what went into that is representing it as a plate. So the idea here, looking at this plate, if you have this much of your plate taken up by fruits, and this much by vegetables, and this much by grains, this much by protein, and then they have dairy as a cup of milk off the side, you're getting a good balance of the various types of food that you can take in. So it's a good, easy way to kind of visualize nutritional needs based on what's on a plate right there. Shoot, I thought this had... Nope, it didn't. I don't have a whole lot of time to go into this because we're about to wrap up here, but I thought it was in this chapter. It must be in a different book that I'm thinking about. But I want you to look into essential, actually, let me type it here. Essential nutrients, which, oops, too far. There are the essential amino acids, of which I think there are 11 to 13 that you have to get through your diet. I can't remember how many exactly, and it actually varies at different points during your life cycle. But these are amino acids that you have to get in your diet, essential amino acids. There are also, I should put essential here. There are also essential fatty acids that you have to get into your system. And these are commonly referred to as omega-3, omega-6, and sometimes omega-9 fatty acids. Okay. there are specific names of the fatty acids that you might find in reference to foods you're eating, but this is describing their chemical structure. When you look at a fatty acid, it's just a hydrocarbon chain with some other stuff at one end. But opposite the stuff that makes it an acid, the hydrophilic part of it, the other end of the hydrocarbon chain is the omega end. So omega-3 is referring to the third bond in from the end. Omega-6 is the sixth bond in from the end. Omega-9 is the ninth bond in from the end. So so let me just see if I can find a quick picture of course, a bunch of pictures of food. Here we go. Okay, so ignore where it says methyl end here. That means something different. But the omega end is this end opposite where these oxygens are part of the fatty acid. If we go in from here, the first, second, third bond, this is an omega-3 fatty acid. There's a double bond in that position. Okay, an omega-6 fatty acid has a double bond in the sixth position. It's not referred to as an omega-6 if there's already a double bond in the third position. If all of these were single bonds until we get to this point, then it'd be an omega-6. And if the ninth position was the first double bond, that'd be an omega-9. These types of fatty acids are very important for making membrane lipids, phospholipids. So if you don't get them in your diet, it can affect your ability to make the proper lipids necessary for cell membrane. So you need those in your diet. And since they're a big nutritional thing and people are always wanting to sell you various sources of these, you have a lot of pictures showing you sources of them, whether it's pills that you can buy at the store or fish or various grains have high concentration of these types of lipids. I suppose if I change this 3 to 6, we'd get some other pictures. Yeah. So here's another picture. Like this is kind of what I was just showing you a second ago. If the first double bond is in the sixth position, then that's an omega-6 fatty acid. Okay. And the same thing for omega-9s, which are not as crucial. The 3s and 6s are more important, but omega-9s can be considered essential fatty acids too. So, so that's just a quick sort of definition of what those essential nutrients are. I had thought the book covered that in that nutrition section of the chapter, but I was mistaken. If you want to look into a little bit more detail about what the essential amino acids are or what the essential fatty acids are, you can look at that. There might be some questions here or there about that. I can't remember how much is mixed in with that, but that kind of wraps up the material from the digestive system chapter and the metabolism chapter. So I was afraid I wouldn't get through all that tonight. Yay for me. So we'll pick up with the urinary system next week. Sorry, hold on a second. We'll pick up with the urinary system next time, which is 2 weeks from tonight. And I would also like to mention, if you tend to stop by the bathroom before you come to class, I know it's hard to remember this over 2 weeks, but please come to class first because we're doing your analysis. Okay, so if you go to the bathroom first before you come to class, you're going to have a little bit of trouble producing a sample. Have a nice couple of weeks.
Can you just scroll up just slightly to the appointment?
Hold on just a second. Let me turn off the recording.